But I especially want to just give a shout out to Dr. Lover and Johanna and Fatma, who helped to pull together all the pieces of Project Itch that we're going to talk about this afternoon. And I'm grateful for that effort, because we started this without really any staff. And so you guys were doing a great job of helping to get all that started. And so we went some 15 months before we even hired Nolan, our project coordinator. So I'm also grateful that he's finally with us and can take some of these things off our table. So I was trying to think about a title for today. What I want to do is do a little bit of a victory lap, but also kind of update. And I thought, like, update, NEWVEC update, that sounds boring. And I couldn't think of different words. I looked in the thesaurus, so I came up with this one. It's like, oh, it's our cotton anniversary. That's the 2-year anniversary. It symbolizes the strength, weave, and comfort between, in this case, two people. But that's basically what we've been doing is, since we started 2 years ago this fall, we've been building teams, right? We've been working together as teams, and that means getting to know each other, getting to know our strengths, and understanding our aptitudes and all that kind of stuff. And so it's really been a growing process, and I think we're at that point now where it's kind of coming together for us. And the research that we'll talk about today, for the first time, all of us being in the same room, will reflect that kind of achievement. So I remind you that the center has 3 prongs to it. So we're doing the operational research that we're going to talk about today. We're also expected to be doing some training. So we're a little behind in getting all that up together. Dr. Lover's been working very hard. So I don't have to tell people that are in the university the difficulties of creating new programs, but we do have plans for these concentrations in the public health major, as well as a certificate that would reach across different departments here. So academically, we have these things coming together, and I'll talk a little bit more about other training programs in a moment. And also we're doing community practice. So that's a fundamental aspect of what we're doing. Like, communicating the research that we're doing with the practitioners is fundamentally important. And it's the opportunity presented to us by the center that may not happen when we're all working in silos in our laboratories. So training programs, there's the academic ones I alluded to. And to a certain extent, many of you in the back row are, products of that or members of that, participating in it, because we're training you in our laboratories. We're also creating new courses. Andrew taught a course in the spring. That's a brand new course in public health entomology, sorry. And I'm teaching medical entomology for the first time in 16 years. Jane took it 16 years ago and somehow ended up back here today. What I can report about that is one of the reasons I didn't teach it all those years is we offer it on the books, and it'd show up, we'd get maybe 2 or 3 students enrolled, and it wasn't worth teaching. This year we have 17 students enrolled in the course. So I think that reflects interest in the course, interest in the topic, and that we're trending in the right way. Training programs also are extended, like the extension piece of this. We're going to be standing up some boot camps. We've basically supported the efforts of other COEs like NEWVEC by supporting travel grants to people go and do those, but we're clearly going to be doing that ourselves. We've been working with the mosquito control districts to do training in mosquito ID and also tick ID. We're going to continue to do more of that in the coming years. One of the things that we wrote in that original proposal that is still getting off the ground was we proposed that we were going to create a network of affiliates, and we called it ParkNet. That was actually Michael Wright's idea. Michael Wright, the grant writer that was working on this with us. Partners Allied in Research and Control of New England Ticks was the ParkNet idea. And we talked about the need for embracing these people in the community practice, but we didn't, like many aspects of the research, we didn't necessarily work out all the details of what that was going to look like. So we're still sort of uncovering that process. And what I'm starting to see now is that probably the affiliate network is more of a set of affiliates or affiliate networks, that there are multiple different affiliations that we can imagine. And I'm just thinking about the ones from particularly our engagement with the Itch project and the Project Whitetail that you'll hear about this afternoon. Is we have, for example, the PCOs that we've been working with. So Fowler and Son and Bartlett Tree were the units that we worked with on Cape Cod for the 170 households that we sampled in Itch in 2024. And they were very helpful in that they gave us customer list and connected with us. They seemed to be partners that want to understand the utility of these different treatments. They seem to be on the right side of the equation. We have the land management, like the trustees of reservations and Sheriff Meadows, that provided us with access to the sampling, the properties that we sample out there. And of course, then we have the residents, which is, I think, a very important piece of our outreach mission. All the people who allowed us into their backyard, they are in essence part of an affiliate network as well. And then of course, the agencies that we're looking to build on the strengths that they've already put together, and to a certain extent, the affiliations that they already have. So I've said from the beginning, I think there's value in partnering with state, county, and university extension. We're still having those conversations. And then we have agency partners like Martin Fian, who's from Mass Wildlife. I'm going to tell you a little bit about how that affiliation has plugged back into our research. And you guys can get more details from Martin this afternoon. So what I want to tell you about is a little piece of little project that really was the baby of Martin and Pat Pearson, the postdoc in the laboratory. So we had this idea, we put as one of the planks of research that we wanted to explore host-mediated intervention strategies, in particular, looking at white-tailed deer. And you know that Scott Williams is doing some of this stuff at Connecticut Ag Station. We thought that it would be valuable to be able to have a captive deer population and a deer enclosure where we could do research on have a test bed basically for the different interventions. So Pat and Martin worked with abutters and agencies and municipal conservation commissions and IACUCs and people on and off campus to put this facility together. And I'm going to show you a video of where it is. So it's about 1.5 miles from this campus. It's a spot of woods that's owned by the University of Massachusetts. It's private land on either side. That's the green grass parts. And that's my truck right there. And this is the road that the farmers are using to access the either side of the property. And we carved out a 5-acre plot, and we basically built a fence around it. The deer are recovered, they're bottle fed, they're not going back into the wild, so they can come to facilities like this one where they'll be used to use those to test those different oral therapies. 5 acres, basically 2.5 acres on either side of that clearing in the middle. And the setup is we have a large fence, the 10-foot fence. I'll show you pictures of that in a moment. And then there's an enclosure within an enclosure where the deer spend the first part of their life. So it's not built yet in this picture, but the enclosure within an enclosure is here. So the deer, in this case, arrived in July. They're bottle fed twice a day with a team of 10 undergrad students from vet and animal science and natural resources. This is another approach coming from the west. The same plot of land, that's the green spot you saw before. This is just the shot I did. That's my truck again. I took my dog out there and did some drone videos before the fence was built. This is where the enclosure lies now, the internal enclosure. And then there's a sort of an antecedent or anterior room that logs on there. There's the shelter that they rest in. Here you see the road that approaches the facility. This is coming from the main road. It's a gated road, so we have limited access to it. And what you see is the fence. It's a 10-foot fence, which is built incidentally to keep deer protected safely inside. Wasn't built to keep things like bears out. And so about a month into it, when Pat happened to be in Spain, we had a bear climb over the fence, get inside, and climb out the other side. And so eventually Pat built an electric fence that surrounds the whole facility to protect the deer. This is, I think, about the first week after it had all been built and before we actually had the deer. I don't have any pictures of the deer this morning, but there are 3 deer in there. We expect to get 3 to 5 each year for the next couple of years. And they're housed initially inside that bladder enclosure. And then after they are weaned off the bottle, they go out into the wild. And the cool thing about this is, unlike other facilities that are doing this kind of work, we don't think there's going to be need to attach deer to or attach ticks to the deer, because Pat's already finding that they're naturally acquiring the deer from their habitat. So it's unique in that aspect that it's deer in their natural habitat, acquiring ticks in their natural habitat. And so the things that we will try, and I'm purposely being a little vague about that because we're still working out exactly what those are, but those will be applied to treatment animals, and we'll be able to test them for ticks economic. I finished with this slide, which is the slide I've been showing since the beginning. When we got the center and I'd go around and be talking about it, I want to just emphasize that everything we're doing, we're still very much not written in stone. We're still in a whiteboard phase. So we're still open to a suggestion for how we can be doing things differently. And look forward to hearing everybody's input. So.
Well, thanks everybody for coming. I wanted to talk a little bit about some work that we did for the last 2 years. I've done it before as well, and I'll give you some background on it. But she couldn't be with us today, but this is our newly minted master's student, Chantel, who undertook most of this research. Anyway, she's now back in San Diego, so she couldn't come. I think we all kind of appreciate that we're sort of in this field of looking at residential tick control, and that the people at the home side of it all, their mailbox is flooded with things like this. They don't know what they're spraying to treat for ticks. And even after you ask the companies that send out those colorful flyers, you don't exactly know what they're treating for ticks. But generally speaking, the big selling point is natural products, minimal risk, not needing to be approved by EPA. And because of that, we don't really know how well they work. And so I realized this years ago, and with support from CDC, we created a project that would sort of in a very robust way try and assess and screen natural products for their efficacy against nymphal stage black-legged ticks. And this is just what that looks like. And our field design, you can see we're interested in knockdown and residual efficacy. So knockdown is sort of spraying on the ticks that are in there, and then the residual is done 2 weeks later. So a 2-week residual, we've sprayed the plot, but then we add ticks to that just to see if it had any residual activity. And so all of these are individual treatments. We replicate everything 5 times. We're able to use 60 ticks. So it's pretty robust. Like all of you sampling in people's yards, you realize that if you'd have to do a lot of work in order to get a statistically sound assessment of treatments. Maybe the main team has done a better job with that. And I know Nita has some success too, but it's a lot of work. This is a lot simpler. So we've looked at it this way. Is sort of the sampling plan. So we deploy the ticks the day before we get started. We apply the products on day 0. Then we sample on these 3 days for the knockdown, and then we deploy ticks again at the beginning of the residual period, and then we look at a 2-week residual efficacy as well. This is how we do the sampling with a sort of a customized little flag. And the way it works is you sort of pump it up and down for 10 seconds, and then you look, and then you take the ticks off, and then you do it again until you don't get ticks for 3 sample times in a row. And then you can move on to the next thing. It's a little bit tedious, but it's not that bad. You get to sit on a little stool in the woods. It's not too bad. To get to this point, though, we have to raise ticks. And so for our project, we probably raise 10,000 nymphal ticks every year for deployment in the woods. And it's a big job to raise that many, but also the day that you're getting ready to put them out, you have to count them all out as well. So sort of 4,000 or 5,000 ticks to deploy. Takes a lot of student help. And then we've been blessed to have a lot of good students that have helped us with this in the summer. So this is putting ticks out. You just sort of knock them into the arenas, tick habitat right on the, similar to what's on the edge of people's yards. And then we cover them. It's not to keep the ticks in, but it's to keep animals from taking our ticks out accidentally. Then the day of the spray, we mix the sprays up in these little vials, and we have a device that's powered by CO2, and it puts out a fairly typical spray in this small area. We have this spray shield that just sort of keeps it all there, because you could see that everything's sort of close together, and we don't want any cross-contamination, because we sort of mix within the array. We mix the different treatments, and so we have all these flags. It gets a little complicated for a guy like me to try and figure out what I'm sampling, but that keeps us kind of dumb to the whole thing. You don't know exactly what's in there until you sort of break the data down later. This is what the sampling looks like. And this is a team that was doing a study. And so the interesting part is when you sort of accumulate all of the data and sort of analyze it. And this is the first year of the first contract that we had with the CDC, and the green products are all natural products. And then water is one of the controls, and Talstar bifenthrin is the positive control. And what I hope that you don't have to look at all of them necessarily. I hope that you'll appreciate that some things work really well and most things don't work really well. If you remember the CDC study that was done that showed, I think Nita was part of that as well, that if you had achieved 60% tick reduction, you still didn't really impact people's tick encounter rate or the level of disease prevention. And so 60% seems to be not good enough for residential tick control to be very valuable. But you can see that bifenthrin, in our case, always gave pretty good control, and some of this natural pyrethrum actually had pretty good knockdown control and a little bit less residual. But one product, I'll get to this in a minute, NEWVEC, we've all heard of it. Most of us can't say it right. This was an early formulation that the CDC had that was derived from Alaskan yellow cedar. It's also now synthesized as a food additive and being potentially prepared for marketing as a tick control. And you could see it gave pretty good knockdown control in this particular early phase. This was back in 2012 to 2015 that we did this work. Then we had another contract in 2020 during the pandemic from the CDC, but we were able, since it's outside work, we were able to do that. And I learned not to put the product name because some people take offense when their product doesn't work. And so I just put the active ingredients here. And you can see we had granular products, we had liquid products. But again, I rank them toward sort of in terms of efficacy. And you can see most of them are not really ready for prime time when it comes to tick control. Here are the controls down here, the positive controls, all with high kill rates, pretty good residual rates. But the best one in that group had pyrethrin in it as well, which is natural, but still not that great. So I think that a lot of these are all commercially available products, and this is what people are getting and putting on their yards, and this is likely the level of control they're getting. So it's a little bit problematic, I think. Well, so I wanted to talk a little bit about NEWVEC. So CDC had a particular effort to move this along, and there was a Swiss company called Evolva, and a few years ago, the EPA did approve NEWVEC as a pesticide, in particular as a mosquito control repellent, and also there was some interest in developing it as ticks. But I think our work is probably one of the first ones that is using the Evolva product to test it its efficacy against ticks. And so here you can see sort of an array of the information that we got from that. So we got 3 products from them. We called it Group A, Group B, and Group C. And we got it in 2 concentrations, 10% and 3%. And Group A, I think, was designed to be might have been a surface spray. Group B was supposed to be a repellent, and Group C was sort of mixed as a yard spray, like a hose end yard spray kind of a thing. And so you can see that it's dose dependent. The 10% generally was much better than the 3% ones. And Group C and Group A both achieved knockdown kill rates pretty comparable to the Talstar right there. One thing you might notice, this one called placebo, we weren't really told what was in it, but we knew that it didn't have nootkatone, but it looks like whatever they put in there also had some killing as well. One of the exciting things that I think we did, so we also have just in the last few years started getting Lone Star ticks in Rhode Island, and so we wanted to see if these products might be effective against Lone Star ticks as well. But any of you that know about Lone Star ticks, they're far more aggressive than black-legged ticks, and so we figured they were going to want to crawl out of our little arenas. The black-legged ticks, pretty much most of them end up just staying in the little arena. But so we needed to devise a system where we could potentially contain the Lone Star tick nymphs inside. So we sort of made a double-tiered arena, and we used some sticky stuff that would try and help keep them in. We didn't really know if it would work or not, but that's what a good graduate student's supposed to be there doing, trying to figure stuff like that out, right? And so here's sort of the base arena down here, and then sticky stuff started up here. Of course, we had to take them off when we did our sampling because it would make the flag all sticky and everything. It's a little bit of pain, but you can see some of the nymphs, and it kept the nymphs in well. We found probably less than a handful in the sticky stuff, that they would go up to the sticky stuff and then turn around and come back down, I guess. So it actually worked better than we expected. We had raised about 1,000 Lone Star tick nymphs for this work, and that allowed us to have enough to have our positive and negative controls, and also to test both products from Group A and Group C. And we just did the 10% there to see if it had any efficacy at all. And you can see the results are shown here. And it seemed like it had reasonable knockdown efficacy even against Lone Star ticks. So that's good. The Group C, which was the one designed for the yard treatment, gave about a little over 90% control. The last thing I wanted to talk a little bit about, we had a group at URI, Steve Alm and Matt Kieswetter. He's a chemist, and they were interested in a problem of varroa mites affecting honeybees. And so one of the big issues with backyard and people wanting natural products is they think they don't want to kill honeybees. And so it'd be nice to have a more bee-friendly tick killer. And so Matt, as a chemist, had this brilliant idea. This is a varroa mite on a honeybee. They had trouble sourcing, the reason I got involved, they had trouble sourcing varroa mites, and they thought, oh, nymphal stage ticks are about the same size, and I have lots of those. Could we just use that as a surrogate? And so I said, that seems great. They hadn't thought of this whole bee-friendly tick killer thing, but I thought, well, while we're giving them ticks, we might as well figure out if we can see if there is an effort there. And so what Matt appreciated is when people develop insecticides, when these companies, they actually are looking for broad spectrum control, right? That's where the market's going to be. Let's get something that kills. And they do these various screens with all kinds of organisms, and they actually toss out the ones that have differential control. But we're more interested in differential control now. And so he had student who synthesized some derivatives of fluvalinate. Fluvalinate is a synthetic pyrethroid, and so she came up with several. She made 5 initially, and we screened them. So this fluvalinate, TF there, is the fluvalinate structure, and then these others, they're called KP because the student's name was Cassie, and her last name started with a P. And so we did trials in the laboratory to figure out a lethal concentration 50 dose, and we did that by looking at, screening a whole bunch of concentrations with 10 ticks per vial and replicated 4 times. And we ended up with 2 products, KP 41 and 75, that seemed to have a fairly good differential kill, less toxic to bees and still killed ticks. What I can show you here are the results from the lethal concentration 50 work. So you can see here's fluvalinate. Here it is against bees. You can see there's a pretty good difference between the kill rates on the concentrations. And this was, both KP 75 and 41 appeared to have reasonable differences, but the KP 41 actually, was 20 times less toxic to honeybees than fluvalinate, which is, I'd say that's bee friendlier at least. And so it was still toxic to ticks. And so we needed to see what happened if we put them out in nature. And so we did that in the second year of Chantel's study. We tested the KP products at both 0.1 and 1 milligrams per mil, just to see. And you could see, again, there's a dose response. The 0.1 seemed to be much better, but all of the KP products seem to have reasonable tick control capacity, at least, when compared to both this Maverick, which is the fluvalinate product that's available for spraying backyards by PCOs, and Talstar. So fairly comparable results. In moving forward, I think we have been able to say that the 10% Nucatone seems like the way to go. Company and discussions with them would prefer thinking about the 3% one because it's more cost effective to sell to people. I guess it's expensive at this point, but it was certainly a little bit less effective than the 10%. And the LD50 for the KP75 is not all that different from the LC50 against bees, but the 41 is got a almost 20%, 20 times difference in terms of its efficacy against bees, but the efficacy is a little bit low for the lower dose one. It was only around 60%. So we may have to make suggestions going forward that we could maybe either look for other products, and I know they're developing some additional things to test, but also just work with the concentrations to see if we could boost it up to get a little bit better tick killing without harming the bees. So that's where Chantel left us with this work, but I think it was pretty informative and hopeful. I'm a little bit hopeful that we can move forward with some actual natural products that are good tick killers.
All right, well, it's good to see everyone. My name's Bill. I'm in Vermont, and I'll tell you about what we're up to over there. This is the team here, and Veronica, and I don't know if Mark is here. No, they happened to visit us, so I thought I'd include them in the photograph. And this work is also being done with Kristen Ross, who is a faculty member at Vermont State University as well, a restoration ecologist. So I'll just talk about the situation in Vermont in case you're not familiar, and then very briefly on our version of Project Itch, very brief, and then the vegetation management, which has been the focus of our work last summer and going forward. So in Vermont, we have some of the highest per capita rates of Lyme disease reporting in the country. When you see a number 1, this is the CDC data by year, and the number 1 means we were number the highest per capita incidence, so not number one in a good way. The data, as you probably are aware, after 2020, 2021 kind of falls off, and there's probably an updated graph. The maps up there are just showing over time at the county level how the per capita rates of reporting vary by county. I'm in Rutland County. The study sites are highlighted in the squares there, and I just put this together for this talk, the three maps, and things look similar over time, but maybe changing a little bit. Rutland County is not that dark color anymore, so things might be shifting around a little bit. But we have a lot of ticks and a lot of cases of Lyme disease and anaplasmosis and babesiosis. I'm starting to realize is a much bigger problem than I've been realizing. Just talking to people from my area, I've talked to multiple people who've had it, but maybe because I'm a tick biologist, I get that kinds of feedback. But we have babesiosis for sure. I've been doing some long-term monitoring at several sites in Rutland County. This is just one of them, and I haven't put on the 2024 data yet, but I guess the take-home message here is that things fluctuate every other year. They alternate. And there's just a paper that came out saying, hey, Lyme disease risk is lower in even numbered years, and that's consistent with our tick data. 2024 was low, but possibly one of our lowest years. I haven't put it on yet. And Dave Allen at Middlebury College has the same results for Addison County, which is a little bit further north. And he was able to connect this back to the host populations as well. So they're probably, and this is Dave Allen's data, it's unpublished, but I don't think he'd mind me telling you that they seem to be tracking their food source, the white-footed mouse, for example. So really briefly on Project Itch, I had a student from my lab work with Jason Hill, who's at the Vermont Center for Eco Studies on the eastern side, almost due east from where my sites are, in the Dartmouth area. So he's working in New Hampshire and Vermont. And what he's doing is testing, he's following the PCOs who are going in and spraying with essential oils. That's the big product on the eastern side. We don't have that happening in Rutland County. And he's going in before and after they spray and measuring tick densities as well as the effects on insect communities. So this is a beat sheet right there, and you just whack the shrubs and collect the insects and see what you've got. And then he's doing some dragging as well. I was shocked. I have never given much thought to dragging on lawns, but the day I was out, he was finding lots of ticks on the lawn. And I guess that might not be a surprise to many of you, but I didn't think he was going to find any, but he did. So that's just a brief on what Jason's up to, and he will be. I think he shared some of the data in an earlier talk, and he'll be releasing papers over the next few months and years, I imagine. So our focus in Rutland County is on vegetation management. We thought that this was a good fit for how landowners are likely to respond to ticks. We have very large properties. It's very common for someone to have 10 acres or 30 acres, or more. And people are very intimately connected to their landscape. They're using their land, and they take things into their own hands. But vegetation management seemed like a good fit for what people might be able and willing to do. And as if you're not familiar with why vegetation is important, I'll give you a brief summary. So we're interested in understanding the link between invasive plants and ticks, and which ones might be associated with tick populations, and can we reduce tick populations by managing vegetation. So on the right is an infestation of Japanese barberry. That is it's a relatively small area, but that's about as much as we see in Vermont. So that's a high infestation, but most of the state, it's still fairly new. And then we're also looking at some native plants, and the best native plant we could find that has the similar sort of density as barberry or honeysuckle are native ferns. So that's why we're looking at those as well as possible habitat for ticks. So Japanese barberry, this is what it looks like. Another shot of that infestation. If you go out now, and there are these nice, pretty red berries. And this is just a plant that is sort of free for the most part from herbivory, so it can spread very quickly. At the same time, they're filling the space where deer are browsing native plants, right? So there's a forest health issue here. And there's been a lot of research, especially in Connecticut, on the effects of barberry. And I pulled up this study by Scott Williams. Just the graph was just pretty straightforward and showing that when you remove barberry, you see a reduction in ticks. And if there's no barberry to begin with, you have a few ticks as well. And what Scott is finding is that the ticks seem to like the habitat. It could have to do with relative humidity. Their primary host, the white-footed mouse, likes to hang out in barberry as well. So there's easy transmission of, well, there's a food source there, and then transmission of Borrelia burgdorferi, the pathogen that causes Lyme disease. We're also looking at Japanese honeysuckle in general, not just Japanese, the bush honeysuckle. And this is another invasive plant that is believed to propagate tick populations for a different reason, because it's the deer tend to like this habitat as a refuge. So when you get these high densities of honeysuckle, and this is some work that Brian Allen did in the Midwest on the Lone Star tick. And when you have these high infestations of honeysuckle, you tend to have lots of deer, and the data here is the dung clusters of deer, and then high rates of infection with Ehrlichia. So this is just an example of one study on honeysuckle. So we have these two plants in our study sites in varying levels. And so we set up last summer I'll go through the experimental design. And you can tell me if we went overboard or not. We were trying to be as unbiased as possible. We didn't want to just say invaded and uninvaded. We wanted to have an unbiased look at the different sites and a way to quantify. So the first thing we did is we ran some transects as much as we could to get the area, a representative area of our study sites. These are all private land, so we were constrained by property boundaries in many cases. And so we ran these transects and then looked at the vegetation using a belt transect, 1 meter, 2 meter long belt, and just every 10 meters stopping, and the students would rank from 0 to 3 how infested the location was with these invasive plants. Okay, so the reds are invaded, the lighter colors uninvaded. And we have these 4 sites. 2 of them are managed, and it's by the landowners, just out of our control before we even started. One was in this case, this is where that heavy barberry infestation is. This is another property, another owner, and then this owner had someone, in fact, a former colleague of mine who's a plant ecologist, pull out, he had his sons go in and pull out their barberry. So we have this natural experiment, high barberry, no barberry. Up there, this one up there, the owner sprayed with he had a lot of ticks, and he just decided to spray his barberry with glyphosate, and he did cut the ticks down. And then on the left is another landowner who has not managed, right? And then we have these 2 unmanaged areas. So we got the qualitative vegetation data. And then the next thing we did, and this is just one example, we got the qualitative data. Then we went in and set up research plots. We found, okay, for example, this is an invaded area, so we put some research plots in there. Higher up, we found an area kind of to the left there that low invaded but high in ferns. That's HF, so we put some plots in there, and then a lower invaded area further out there. So using the qualitative data, we set up these research 3 meter by 3 meter plots where we would really comprehensively look at the vegetation, characterize everything, and then do flagging for the ticks, right? And so 3 meters by 3 meters was like the largest area we thought we could reasonably do to do this work. And so we repeated this at all 4 sites. They didn't all have, usually we had at least two areas. So instead of like 3 clusters, there might have been 2 at the other sites, if that makes sense. This is just another example of what I just explained, where we had the qualitative analysis, and then we put in plots. High fern. This is an interesting site because this site has no invasive species. Like literally, it's like so healthy looking, although maybe not as much understory either, because there are a lot of deer here. So high deer, low invaded, and it's our tickiest site. In fact, of all my sites, this is now the tickiest, so go figure. So we can at least look at ferns here and see if that's doing something. And this will be interesting to track over time. Are we going to see an invasion of invasive plants over time? So what you're looking at is the different sites. So site one, the high invaded area, there were 2 high invaded areas in site one. It just shows they're organized by high invaded, low invaded, and fern. And the results are not surprising, right? The greens are the honeysuckle and barberry, and we included buckthorn in there. And then the purples are the fern. So the pattern it's not surprising, but there is a range. So it's not clear cut, like totally invaded, totally uninvaded. Is that data going to matter? Well, we'll find out. We also have other plant data I'm not showing here. So we can bring in, we have everything in the understory, overstory. So we could possibly do a more like a community signature if we wanted to. I'm not sure if it's going to show much, but we have that data. And then we did the flagging. We're changing. So we're going to try out a new flag this summer. It's a heavier cotton. It's like a rubberized cotton. And because it's really hard in the thick barberry, we need something a little more durable. So we're going to try that out. I almost shouldn't show you this data because it's too early. We did this summer sampling. We had to set up all the vegetation plots. Then we started sampling. We were past the nymphal peak. That said, and the numbers were really low. So you're seeing like high invasive, you see more ticks, right? High fern, low, these are low invaded and not much different. So you might say, yeah, those 3 follow the pattern, but it's a very limited data set. And then this one, of course, does not fit the pattern, right? And so we just don't know if that's meaningful or not. It was pretty late in the season. We're still also getting the baseline data for these sites to know like how many ticks they have in the first place. And so, yeah, I just want to acknowledge this. Based on our very limited data, we're not seeing the pattern, but I'll tell you what happens this fall. So we're going to go out this fall, and so these are the next steps. So we're going to start sampling. I have to go out tomorrow and get this started. Tom made me feel a little bit better that maybe I'm not too late yet. I got worried that I was getting late with the adults. So we'll flag and we'll see what we come up with. We'll do the same. So we have these plots set up. So we'll flag again next summer and next fall. Then we'll have 2 years of baseline data, which my long-term data set, that suggests you need at least 2 years of baseline because the populations fluctuate. And then we'll try some sort of intervention where it might have to be a limited to begin with, but remove some plants, and then we can track that over. So this is year 3, right? So we can, at the very start of year 4, we can manage, and then we'll have year 4 and 5 to track the results. So that's what we're up to, and just some people to thank, and I'll take any questions if there's time.
Okay, so good morning, everyone. So in Western Connecticut, we have been focused on backyard prevention and understanding risk for tick-associated diseases, specifically those associated primarily with black-legged ticks. So if you live in this region, you're probably familiar with many of the common recommendations to prevent tick exposure in the residential landscape here in the Northeast. But CDC's tick prevention website has like a whole myriad of backyard strategies that they highlight to help keep homeowners safe. Either two of the major ones include creating a tick safe zone, using like landscape modification, and also, of course, applying pesticides to manage tick population. So when we think about this recommendation, mostly the application of pesticide recommendation refers to the broadcast application of tick killing pesticides. So traditional pesticides, particularly synthetic pyrethroids, have been pretty well studied, and despite being commonly recommended, we see that in Connecticut, where we have, of course, lots of ticks and we're highly endemic for Lyme and other black-legged tick-associated diseases, that only 21% of residents apply pesticide for tick control. And that's in contrast to some of the other measures that they will do, like more than 50% will treat their pets or perform tick checks. And so while they didn't measure why in that particular survey, in another survey of residents, 61% cited safety concerns as a main reason for not using pesticides. And so in addition to this like chemophobia, homeowner decision making when it comes to backyard tick control may be further, complicated by the fact that there's so many different products that are available to them, not just including like the pyrethroids like bifenthrin and cyfluthrin, but also there's the pyrethins. Some of you may be familiar with entomopathogenic fungi, rodent-targeted devices. Those are your bait boxes and your tick tubes, as well as natural products, which may contain botanical ingredients that are more increasingly flooding the market in our area. So in Sarah Niezebecki's TickNet survey, she also reported that 14% of Connecticut residents reported applying natural pesticides. And although we don't really know what they applied, we're not really surprised about this, because we see in our area, we've been seeing an increase in a lot of these professional pest management companies offering natural tick control. In some of these cases, it's the only type of thing that they offer. So over the past several years, we've surveyed the licensed pest management professionals in our state about their tick control practices. We found in our 2021 TickNet survey, we found that more than half of them reported offering both synthetic and natural pesticide options, and nearly 20% reported only offering natural options. And then in a separate survey, we saw that our respondents, there it is, reported, nearly a quarter of them reported applying a cedar oil or cedar oil based product as their primary tick control service. And so the increasing use of cedar oil based residential tick control has been a pretty great interest to us. We know they're offered, we know they're marketed as safe, which can be appealing to the homeowner. They're exempt, Tom alluded to this, from EPA registration as a minimum risk natural product. And unfortunately, right now, we don't have a lot of information to talk about their efficacy. So in the beginning of 2024, we launched our Connecticut version of the itch project. And for this study, we started out by just asking, do properties treated with a professionally applied cedar oil product have fewer ticks than untreated properties? And so we recruited properties early in the year. We ended up enrolling 46 and sampled them between May and July. We visited them each 3 times. Half of the properties were treated professionally by a commercial PCO company with a cedar oil-based product, and the other 23 had not been treated. So in the interest of time, I'm not going to show you all of the data, but I just wanted to point out some of the sort of the most important takeaways. So first, when we looked at the overall tick abundance, you'll see that the tick abundance was similar between our treated and untreated properties. And even though maybe slightly less at the cedar oil treated properties, we did not find a significant difference between them. And in fact, though, when we break down our findings by the products applied, you can see that there's, quite a bit of variation between them. And of course, our study was not really powered to evaluate the different products, but hence is what we ended up with because of the, many complications of recruiting and controlling all these variables. So, but seeing this, we really just came away with more questions. Like, first, what do we know about these products? And Tom shared a little bit about these in his presentation. And actually, the only two of our products have been studied, and they were in Tom and Megan Dyer's study at URI, and they found that both the cedar side and the tick kills had very limited knockdown and residual control of black-legged tick nymphs, and neither product was significantly different from the water control. And there's no other published data for these other products, the Cedar Cure or the mosquito-free or tick-free products. So our first year of this study, was also limited by variation in the different application methods. So as you can see in this graph, the properties that were treated with cedar side had a significant amount of ticks, but you can also see that those were the only properties that were treated with a backpack sprayer, whereas the other products, the tick kills and the Cedar Cure, were applied using a high pressure truck mounted sprayer. And we know from a recent study just published by Scott Williams and Megan Linsky in Connecticut, they found that a high pressure application with synthetic pyrethroid worked better than low or medium pressure application with a backpack sprayer. So that could have also impacted our data. So this was just our dipping our toe into this topic because there's very little information out there in the real world about what people are doing and whether it's working. And so although this year was just our first step, obviously more study is needed. So we need to, of course, better understand the impact of the products used, the application methods, and the timing. And we're hoping in this next year we can sort of try to get into some of that. And also some of the human dimensions around using these minimum risk products, particularly cedar oil, the motivations for using them, and maybe how that might, and their perceptions and how that might be able to be impacted by our study findings. So returning back to this slide, I wanted to just spend the rest of this talk just really briefly showing you some of the other work that we've been up to. And so I'm just coming back to this slide because we were talking about pesticides, but there's also the part about understanding the landscape modification. So here's CDC's tick prevention websites, recommendations, and these are modifications one can make to make their yard less hospitable for ticks. And so at least these first four really talk about trying to mitigate ticks and hosts along the wooded edge. And that's because we know in the backyard landscape that most ticks are found in the forest and then in the area 3 meters from the forest, and that can include maintained lawn areas as well. And so naturally, this area is a focus of many of our recommended tick prevention measures. So of course, anyone studying backyard ticks knows that there are a lot of challenges, and in particularly, we have high meter by meter variation and patchiness in the distributions of ticks. And we also have a high variability over time, and that's not just across or between years rather, but even within years and within seasons. So here's just an example of that. I know Bill brought up some of this as well with the interannual variation, but this is our data set. This is for 13 years. We sample weekly for nymphs from May to August, and you can see a lot of variability, not just year to year, but also week to week. And so this, of course, can impact our studies greatly. And sorry, so I don't know if Lars is on this call, and Tom, probably we all want to forget this data, but I wanted to bring this up as an example because we spent a lot of time not that long ago working on a backyard integrated tick management study in Connecticut and Rhode Island. And what I want to point out here is the years 1 and 2, which were our treatment years in both states, had very low tick densities. And so it made it very hard for us to be able to evaluate the impact of an intervention. And this was a 4-year, very not the cheapest study to run. And we really were disappointed because no matter how much planning you do and well-executed study design, sometimes you can't just deal with what the variation can just really monkey with your study. So I think maybe I'm a little bit jaded after 20 years of studying ticks in backyards and having these unexpected findings. And so I just wanted to add, because of this variation, we see some of these other issues, like we don't know how many ticks were there to start with, right? We can't control anything, as with many ecological studies. We can't put ticks in people's backyards to enrich them, right? Like that wouldn't be right. And so to conduct, powerful studies, robust studies, we need a lot of money, a lot of time, a lot of labor. And of course, then we also have to count on being able to recruit the properties that meet our study criteria. So really quick, because I don't have a lot of time, I just wanted to introduce you to an approach we've been taking now. And Sandra, my student, will be talking more about this later. We wanted to know if we could simulate the backyard edge using enclosures. So obviously you heard from Tom, the use of enclosures or arenas they're not new. This is something that's been doing, I think Nell is using them as well, but mostly they're erected in a forested leaf litter kind of area. And so we wondered if we could sort of simulate the actual edge, that high risk area in backyards, then we'd be able to better study, the dynamics of tick activity at this location, either with or without interventions. And maybe, ideally, can we also start to understand the human tick encounter risk if we were able to control more of the variables. And so I just wanted to tell you about last year, we started this tick garden project. And in this project, we tried different designs of enclosures. The one on the left is a modified crop cage with a wire mesh top. And it was only 4x8. And then we also used a 1x16. These are in feet, sorry. Sort of shorter track. We called it a racetrack. And we had infested them, and we're able to track ticks that were marked, either moving from the forest into the lawn and vice versa. We could track their survival and their behavior as well. So we basically wanted to know, are we getting the ticks? Are we able to see them moving in this environment? In our first tick garden design, we could see that ticks were moving into the forest from the lawn, but less so than ticks moving from the lawn. Sorry, I said that the wrong way. Ticks are moving from the forest into the lawn less frequently than they move from the lawn into the forest, but we actually saw that they weren't going they were kind of stopped at the end, maybe because it wasn't long enough. So in our second one, our second iteration, we actually made a longer track so we could see them go. And what we found was that, yeah, they don't go that far. They usually go about 1 meter from where they were deposited, which we kind of know from the literature anyway. But we wanted to know, will these ticks move in our enclosures? Yes. Will they survive? Yes. Can we mark and recapture them? Yes. Can we recover them in a reasonable way, like the amount of them? And yes, the answer is yes. So can we simulate it? We think yes. So the ticks, anyway, we were able to monitor the microclimate too. Sorry. So anyway, basically we've now that we know that they work and we can use them, we are using them to better understand our vegetation management questions. And we're starting with the recommendation in the fourth bullet here, which is mow the lawn frequently. So what does that mean? How frequently? Nobody knows. So, and so this mowing issue is of greater interest to us lately because, and I don't know if you have this in your area, but we have a lot of people who are very interested in either being part of a pollinator pathway, which may include No Mow May, and just people are being part of No Mow May anyway to try and promote pollinator health, increase biodiversity. And so we don't know what impact this will have on our tick populations. And so we are able to study that. So I don't want to tell you what we found because you can wait for Sandra's lightning talk this afternoon. So thanks to everybody. It takes a huge team of people to make things work.
So hi, I'm Jeff Garnis. Very nice to meet some of you who have seen many hours on Zoom screens and some of the other folks here in the audience that I haven't seen before, but it's great to be here. So I'm just going to give you a little bit of an update on what we've been doing in New Hampshire for the last 2 years, and I'm gonna tip my hat a little bit here. Well, first mention that my background is very much not in the area of ticks or vector-borne diseases or anything, so I'm a little bit of a tourist here in this group, but I typically study the short version is the ecology and evolution and management of novel biotic threats in forest ecosystems. So I study things like the emerald ash borer and beech bark disease and beech leaf disease, and a new one, and a bunch of other forest pests is sort of the niche I've carved out for myself. So I guess I think of ticks in some ways as a novel pest in our area. We have, well, I'll show you some data showing that we knew that the Lone Star tick was around, but we did pick it up in our sampling, which is not common knowledge to a lot of people. And some of the other ticks are moving north and finding their way to the northern forest where they never were before. So I think this fits in. But I'll be honest that I am not a tick biologist by any stretch of the imagination. So this is just some of my lab group, and I realized I took a slide out that removed Casey Coop, who's been the leader of the field campaign for us for the last 2 years. He is a co-author on this little talk here, but we've got a sort of diverse group, and some of these people have helped out with some of the field sampling. And we learned how to do tick flagging. So I'd heard about tick drags for a long time, and then I realized that tick flagging was now the in vogue way to do this, and so we had to learn how to do it and how to make these really high-tech pieces of equipment. And then we learned that they're actually have their own challenges. So there's no great way to sample ticks, as it turns out. Who knew? So I grabbed a screenshot of this is actually my house up at the top there to represent one of the things that I thought was really interesting about some of the discussions we had early on was trying to think of, I'll say one of the things that attracted me to this group was the multi-state effort and the fact that people were trying to get their minds on the same page, even though they've been working across different disparate landscapes and sort of cultures and all sorts of things, and trying to produce a replicate data set across big areas seemed pretty appealing to me. And then the reality set in, and it turns out that that's actually pretty hard. And part of the reason that's hard is because our landscapes do differ quite a lot. So I don't know exactly what Rhode Island looks like, but this is a lot of what we see in my area is like this. So, homes with 5-10 acres of land and a lot of forest interface, a small yard relative to the overall land ownership. And so I would suggest, and my anecdotal experience is that, so I walk my dog through a trail, which you can't see here, back through the woods here to the Great Bay, which is over on the other screen probably, in New Hampshire, and that's where we get our ticks. So I mean, the yard for me is not that big of a deal in terms of tick exposure. It's when we walk through the woods and traipse through the higher vegetation and some of the invasive species. One of my favorite things to do when I take my dog back in these woods, I generally try to take out at least one Japanese barberry each trip. So about 100 years, I'll start getting a hold on that population. But anyway, so landscapes differ quite a lot. And I know that people are familiar with this, this approach, but I'm just going to show some of the data that we collected. It's new to me. It's probably kinds of things you've seen many times, but we collected in different zones, zone A being the yard itself, and zone B being the yard forest interface, and zone C into the forest a bit, which is, I guess, standard across a lot of the sampling that we did. And of course, this is how we sampled. And we were able to get 40 sites in southern New Hampshire, clustered in this southern New Hampshire area, which is Durham is right about there. So you can see they're clustered around where I live. I'd be interested in thinking more geographically widespread sampling, but that's of course logistically challenging, and recruiting properties was not so easy. So we did sample 40 sites in 23 and 24. Well, here's a little bit of a closer look. So you can see there's quite a gradient. Some sites we sampled only in one or the other year. The majority of sites we sampled in both years. We actually attempted in the second year to recruit properties from Bartlett Tree that had been treated so that we could do a comparison similar to what you've seen, but in the end that kind of fell through. And so we just have untreated properties for all our sample sites. And the other thing to note here is, like I said, so in this area and up north, you know, it can be pretty rural, and it can be fairly large properties with lots of woods. When you get into Manchester and Concord, which looks like there's a sort of a non-randomness about the way that we sampled in 2024, but these are much more suburban, even urban kinds of landscapes, so very different. So I thought we got very low numbers, but seeing the numbers that on some of, I guess it's very variable. And we, in 2023, in June and July, collected, well, what's about 300 ticks total, and only about 62 ticks in 2024. And we thought maybe we were doing something wrong, but I think the answer is that's just the tick density at the sites that we were studying. And so that's what I'm going to show you. So I noticed that Nita and others used ticks per hour. We tried to equate that to some sort of distance. It really was a time metric that we used, but I'm reporting on ticks per 100 meters of flagging, and, you know, very, very low densities, but much higher in 2023, which fits the alternate year model for whatever that's worth. And overall, although I wouldn't say, I didn't even do stats on this because I'm not sure we have the data to show it, but at least in 2023, Zone B, that forest edge, was the most, had the highest tick density. In 2024, there were so few ticks that we didn't even really see that pattern. But that comports with, I think, other people's understanding of these systems. So I have a bunch of tables that are probably hard to read, and we don't need to really go into the details, but that's just another look at the same data. And I was interested in what kinds of ticks that we got, and it, probably not surprisingly, it was dominated by the black-legged tick. Our sampling 83% in 2023 and 77% in 2024. I don't know if we were supposed to do this, but we collected all of them. This is a notable one, the Lone Star tick. I didn't know it was present in New Hampshire. It turns out it was not a first report, but the fact that we picked it up means it's got to be around. So I was very interested in the pathogen data that we got back from UMass. So this is really Juan's work more than ours, but I'm reporting it anyhow. So the only ticks that showed any positive pathogen detection were the black-legged ticks, and maybe everyone knows that too, but I didn't. And so in 2023, when we got the most ticks, the pathogen incidence was only 16%, so that seemed relatively low. It was quite a bit higher in 2024, but of course there are fewer ticks, so maybe that's just random stochasticity. 66% in 2024. And you can see the Borrelias pretty well represented in this group here, but we did get Babesia and Anaplasma, or plasma, I guess, or Anaplasmosis and Powassan virus and deer tick virus all showed up in our sampling, which yeah, suggests that there's cause for concern in New Hampshire as well as everywhere else. So not usually in the habit of giving time back to talks that I give, but I think I might this time. But so I just wanted to sort of end really with talking about some of the things that I've been thinking about going forward. I really enjoyed Bill's talk about the invasive species. I've read some of that literature. It seems really interesting to consider. So I would be interested in understanding how habitat and land use type influences tick abundances, given the variation that we have in our landscapes in New Hampshire and certainly in other states as well. I think it is important to evaluate the efficacy of chemical and other controls. I see that that's been a theme that we've talked about here. Feedbacks between ticks and invasives is also a really interesting thing to consider. And then I would be open to talking with any of you about things that we should be doing. Like I said, this is not my primary focus, but I think it's been a positive experience being connected with you guys, and we hope to continue it. So
it's wonderful to see everyone in person and finally connect with all these different groups across the region. I'm going to give an overview of the studies that my lab has been working on in the last couple of years, and then some discussion about next steps and what we're looking forward to. So this is just a little bit of an overview of what my lab focuses on and how we think about our research. And basically, over here you have beautifully constructed randomized trials where you control everything, and you have picture-perfect data. And you get a number here that says, oh, your efficacy is 98%. And then you walk out and you try run a public health program. And at each one of these steps, you lose 10% or 20% or 30% based on coverage or uptake, or people don't like the intervention and don't use it, et cetera, et cetera, et cetera. And you get down here and your effectiveness is 37%. So these are not real numbers. This is just the hypothetical intervention. And so the thing I like to think about is how do we identify what those gaps are and then develop public health programs that can mitigate those losses. Because there's tons of beautiful clinical trial data that has very little, or like randomized trial data that has very little applicability to impacting public health. And so this very often involves a lot of diverse study tools to work with populations to understand what the barriers are to moving this relatively, unimpressive number to something closer to a beautiful trial. Okay, so this is the main project we've been working on. So ITCH has a bunch of different components, and we talked a lot about the tick sampling. Paired with that was, or is a long-term internet-based survey looking at knowledge, attitude, and practice among participants for their knowledge about ticks and mosquitoes and kind of what they do at the residential level to control that or don't, depending on who they are. So this is kind of also looking at risk factors for recent tick-borne disease within households. And then Project Listen is a qualitative study looking at workforce challenges in management across the vector-borne disease professional landscape in New England. And so we'll talk about that a little bit. And then spatial epi of tick-borne disease in Western Mass. So this is building on an R21 that Steve and I put in pre-pandemic. And then we have a bunch of follow-on studies with NEWVEC. And then last but not least is this itch research support. So that's been a lot of the research management and IRB submissions have been a lot of time and energy. And then we're also getting all of our tick data into the CDC ArboNet. And that's been a little bit of a challenge because CDC is set up to get it directly from state DPHs. And so they have like a direct portal. And so we're kind of outside that usual reporting system, but we're going to get all of these tick reports into the CDC database, which hopefully will fill some gaps. Okay, HKIP. So the idea here was that aside from some fairly geographically limited studies, we don't have a great sense of the knowledge, attitude, and practices for people about residential vector control throughout the region. So we wanted to try and understand some of the gradients, both socioeconomic and ecological across the region, to just see how feasible and realistic a lot of vector control at the residential scale is. And so this is about a 10 to 15 minute survey. This is the main landing page. And so this is kind of the general sets of questions. We also capture the household size and whether they have pets and how recently they've had tick-borne disease or mosquito-borne disease, et cetera, et cetera. So there's a really rich data set there that we're delving into. And so again, all of these data are from all of you, and you're driving using the bushes to get respondents in your respective states. So there's been incredible amounts of work to get an absolutely stunning sample size. So we've had 6,700 people who have at least started the survey. Some of those did not finish the informed consent or their modules that they chose to skip, but still that's a huge sample size for this kind of study. Massachusetts and Maine are running neck and neck at the top of the leaderboard. And then we have a little bit further. So the others are a mix of mostly New York and then a couple New Jersey, and then there are people from California and kind of random places that somehow got a link and decided to answer it. So the others, we probably won't include in the final analysis, or we'll do a sub-analysis because whatever ways they answer the questions are not particularly relevant to our specific questions. It's good to see that people outside of the region are interested. So I'm going to give two big overviews here. The question is, mosquitoes are a big problem, ticks are a big problem, and then it's a Likert scale from strongly agree to strongly disagree. And out of the 5,586 people that answered those questions, the vast majority of people for mosquitoes were more in the strongly agree, and for ticks, it's in the top category. So there is some variation in terms of the level of qualitative, semi-quantitative worry about the respective vectors in people's households. And then this is a little bit of a busy slide, but basically these quadrants, or whatever that would be for 16s, is color-coded by which area people responded to by state in terms of the highest. So in Massachusetts, people are more or less equally worried about ticks and mosquitoes. So that's why this top box is shaded. Whereas in Vermont, there's a stronger worry about ticks and a slightly lower level of concern for mosquitoes. And so there is definitely some regional variation in terms of the relative importance of those two. I bet if we did kind of a longitudinal analysis, you'd probably see if there was a lot of Triple E or West Nile reports, you'd see a shift in where that is depending on just people's level of interest. But it does show that there's probably a need for fairly targeted public health messaging. Just saying, watch out for mosquitoes and ticks may need a little more nuance. I mean, certainly COVID has suggested that nuance in public health messaging is really difficult, but we should at least try. Okay, and so Fatma will give a turbo talk on some of those further results from this project Listen. So this came out of the realization that there's very little in-depth understanding of what parts of the vector control professional landscape are working well and what's not working as well as it could. And in some cases, this is especially exacerbated by the different public health systems across the states and the region. And so this is developed as a way to try and probe all of those people who have a lot of really important information about what works and doesn't, and use that to try and develop policy suggestions that can help vector control work better across the region. So it's a combination of focus groups and key informant interviews. So focus groups is 6 to 9 people, or 10 or so you sit around, and people can kind of bounce ideas off one another. You get very interesting social dynamics. And you very often find out things you would never even think about putting into a survey. And that's why this thing is really important, is that you can explore themes that help you develop a subsequent survey to ask questions that are directly relevant. Because we can all guess what are probably the problems, but we talk to people that are doing it day to day. That's not the problem at all. Yeah, and so key informant interviews is a more focused one-on-one interview with someone who has very specific domain knowledge. So Johanna's going to give a turbo talk on all of her initial results from the first couple rounds of sampling, or first couple rounds of sampling. And so quickly, there are kind of 3 tiers of interviews that we're working on. At this top level are the kind of boots on the ground public health or entomology or vector control staff in terms of implementing the programs. This mid-tier is kind of a higher regional or county level position that may have some more supervision or kind of general policy. And then the top level is the state level legislators and DPHs, and that level of kind of higher level oversight. So we're working our way, I should say Johanna is working her way through this very complicated scheme. Okay, and then I'm gonna also talk about spatial case control. So this is a project that was started pre-pandemic. And there's this idea that there's this Pareto distribution, which is 80-20. So for a lot of parasitic diseases, 20% of hosts have 80% of the parasites. And that general rule holds across a lot of different scales. And so talking to clinicians here, there is this idea that a very small number of trailheads may be seeding a lot of potential tick exposures. And the idea was if we could find a way to identify those, and then we would have a nice targeted area to do really intensive tick control. So we're basically enrolling people who have recent tick-borne disease as probes of ecological areas that may have especially high human tick overlap. So these are GPS data loggers. They record GPS coordinates, but they don't transmit. So this was kind of important from an IRB standpoint because people could hand us the thing that they really didn't want to share where they went. We can delete the data. No one had any concerns, which was pretty cool. So we got lots and lots of data. And yeah, so that's all of that. And we enrolled people who had recent tick-borne disease or attached tick from clinics, and then people who had any other condition. So we kind of excluded people who had a broken leg or mobility issues that would impact their ability to collect data. So 634 people concluded the screening. Just under 600 actually did the informed consent and went through that whole process. We got 211 data loggers back. Some of them had kind of corrupted data or were limited data. So at the end of the day, we got 193 person levels. This is 2 weeks of sampling all of their locations outdoors in potentially tick exposure areas. So there's just under 1 million data points. And we filtered out people's houses, directly over their house for privacy issues, and then anytime they're over, in the car. So what do we do with all of this? There's a lot of statistical tests that let you look for patterns in spatial data. So the one we're using is called TAT-SCAN, which basically says if your cases were randomly allocated across the landscape, what would that pattern look like? And then it compares the patterns that you give it against that completely uniform background. And there's a lot of fancy ways it adjusts for multiple testing. And so from this, we can get hot spots and cold spots, which are areas of higher potential tick human exposure versus the baseline, as well as cold spots, which are lower risk. So these are what the data actually look like. So these are a preliminary scan of all of the region. So we did constrain people having to live within a reasonable geographic radius of Amherst to keep the spatial scale reasonable. And so from after all of our data manipulations, we then got this map, which shows the hotspots that we are most interested in. We did not see any cold spots in these data. And so James is going to have a work in progress a little later this semester to go through all of the spatial epi in detail. And following on from that, we identified all of our hotspots. And so in some cases, the actual hotspot that was there was so it's kind of this arbitrary point in space or in the landscape that may not be directly accessible in terms of permitting or logistics, or if it's in the middle of a swamp. So we picked a 500-meter bubble around all those and identified the best sampling location within that radius. And so Vera is going to give a talk about the follow-on studies, which is looking at vegetation indices. So this is developed with Bill's group and Allie's group. We have all his permissions in place, and we're currently sampling at a range of those sites to try and understand the spatiotemporal dynamics of ticks, as well as getting some parallel vegetation indices for all the work in the northern tier.
Thank you. So I'm a disease ecologist at the University of Rhode Island, and for the past several years, I've been working on ticks. I also do research on mosquitoes as well. Mostly focus on the biology of vectors and host-parasite interactions, but I have been shifting into the field of management and control, testing novel controls, and through Project Itch, the questions really started coming up of, how can we better support homeowners as they're trying to make decisions with what should they do in their yards. So not only is your tick control helping, but okay, now what? What do we do next? So the acronym for Project Scratch is a little bit contrived, as any acronym is. So this is really about the development of an adaptive decision-making tool for the protection against ticks and ticks and tick-borne disease, not mosquito-borne disease yet. Maybe in the future. Yeah, and sometimes that works and sometimes it doesn't. Like this photo. Okay, so the decision landscape for homeowners, for any household or even individuals, is actually very complicated. It's a lot more than am I at risk, but most of the people in New England just are at risk already. So they know that they're at risk, but now what do they do about it? And you all know the list of recommendations of what you can do is very long. It's very long. And they want to know, well, which one should I do? What's best? So if I do want to manage ticks in my yard, what's the best way? And I think it's actually really helpful that I'm going last in the morning talks, because you can see all of the nuance that's involved. So even so it depends on who you ask, because there are a lot of stakeholders here: pest control operators, public health, friends and family, right? And also, even those that are highly educated in this topic still will say, well, it depends as well, because there's variability across space and time. There's variability in efficacy depending on the treatment and how you apply the treatment. So it is not as though people they're just not doing it because they're not intelligent or not reasonable or rational. It's because it is a complicated landscape with no quick fix, no easy solution. And that is where I think that we as a community can come in and provide some actual tools. So this is a very difficult decision that homeowners are making. And there are some tools that are available, right? There are websites you can go to educate yourself. There's an 84-page handbook you can read if you want to learn how to control ticks in your backyard. And this is an excellent resource, right? But it's also a very hard ask. It's a very big ask to say, go educate yourself, basically become an expert yourself in ticks and tick-borne disease and management of ticks. And many of us may be expert in just one of those things, and it's still very complicated. So that's a big problem, that in order to protect yourself, you have to learn so much. And it's kind of no wonder that there's gaps in knowledge, because it does get overwhelming. The other thing that you find with these, and they are helpful, but as we saw with comparing the properties in Itch even, that it isn't a great one-size-fits-all situation, right? So the general recommendations for creating a barrier in a tick-safe zone in your yard don't really apply if you're taking a mile walk through the woods with your dog every day, right? So these recommendations are not very highly tailored to individual needs or even to just profiles of different potential needs. That was the other thing to kind of notice. So it's not to at all say that these aren't excellent resources, but I think we need to foster some empathy for the people that we're trying to help here. Okay, so the goals of Project Scratch are to simplify the experience of decision-making around tick control and tick-borne disease prevention. Simplify the experience, knowing we don't want to oversimplify what is the situation, but the process of making decisions, it is complicated, but there are ways to structure that decision so that it is less painful and relatively straightforward from a user experience. The other goal is to tailor these recommendations to the risk profile of the user. And Itch is really helpful for this because we are able to kind of come up with screening questions to say, what are property types and certain do pets or not have pets, things of that nature. The other aspect with the structured decision-making approach that we're using is that we get to incorporate, and this is really the goal of the work that I'm presenting today, is to incorporate the values and objectives and concerns of the people using the tool. So having their, recognizing that people have the agency to say, no, I very much care about the environment. I'm not going to consider at all any synthetic chemicals. Now, this is the person that has to still make a decision in that context and those values. What can we do to help them make the best decision? And then the final goal is to incorporate not only the consumer, let's say, homeowner or household values, but also have that marry with all of the collective expertise that we can provide. Okay, so for those of you that were on the call for NEWVEC, the updates, I said, seeking experts, right? So this is the next steps is that we're going to be soliciting people to really put in some time, effort. Folks that are in this room and others that have really put in a lot of work into figuring out and know the data on the efficacy of a lot of different treatments so that we can populate our decision tool with that expertise. So this is what the PROACT cycle, this is the dream with structured decision making. It is, even in the best of circumstances, can be difficult when you're dealing with managing problems with a lot of uncertainty. Depending on your decision, it's really helpful to have a plan going forward. So you have to formulate what is the problem exactly, put a box around it. This is the problem we're dealing with. You identify what are your objectives, and then you come up with alternative actions that you can take that would have impact on those objectives. You use some decision analysis tools, and then you implement action. That's all well and good. When it comes to the reality for vector-borne disease management or wildlife disease management, any number of really complicated contexts with a lot of issues, that it's not as straightforward. So these are dynamic problems that do change year to year. You have a lot of inability to predict outcomes, a lot of uncertainty and risk. Resource allocation is not always consistent, right? And you have multiple objectives, and those objectives might be directly in conflict. So you could have trade-offs going on here as well. For example, you may want to protect the environment, but you also want to protect your health. And so that's a trade-off that everyone's trying to contend with. And then there is a lot of uncertainty in the scientific research as well. And so it's about how do we still go from problem to solution? And our bridge here is trying to use decision analysis. And it's not about coming up with the necessarily best decision in the ultimate sense, but the best decision that we can make given all the information that we have, given the constraints that we're under with uncertainty and complex dynamic systems. So the type of decision analysis that you do really depends on the type of problem that you have. And there are a lot of different problem types. So I've had a lot of fun the last couple of years learning about all the decision tools that are at our disposal. And then I think, wow, how did I ever make a decision in my life before this? But this isn't necessarily, you can actually use these tools for like buying a car or a house. But they are very useful when you come up with problems that are complex, really complex, or you have multiple stakeholders, different interests. So the class of problem that I'm going to talk about that we're really focused on, because in tick-borne disease, actually, in tick management, we have all of these problems, every single one, but try to narrow the focus a little bit and really think about homeowners and what are the decisions they have to make. So we classify this as a multiple objective problem, that they have, if they're deciding whether or not to treat, they're deciding, okay, well, if I want to enjoy my outdoor spaces, and not get ticks, I don't want to get sick, but I also don't want to impact the environment. I also don't want to spend all my money. So there are multiple things that they're contending with here. So the key thing here is we spend a lot of time on our own before actually going and speaking with homeowners, just conceptualizing what is the problem, what are the right decision tools that we could potentially use, and then we kind of updated as we went along. And so it was pretty straightforward for us to say, okay, well, we know we want to minimize the number of sick individuals. That's public health, right? That's definitely something we're worried about. There are ecological considerations and economic considerations as well. Then we're thinking about, well, for an individual household, what does that look like? And we put the boundaries of this decision. You have to define the boundaries, otherwise we'll just keep going and going to whether or not an individual would undertake a tick control project in their yard of any kind. That's very broad, right? And so we're trying to put the bounds on what sort of factors would they consider in this. All this, it didn't change all that much once we started talking to people, but there were changes that happened after we actually went and spoke to people. What we did this summer, and by we, I mean Alexis White, who's here. She did all of these interviews. She couldn't be here today. She was at a wedding, but you know, hopefully in the future she can present on this information. We used the kind of PROACT cycle, and we wanted to identify the objectives, you know, straight from the horse's mouth, right? What are their objectives? And then we wanted to figure out how did they weight those objectives. And we used long form interviews. Again, Andrew, you did a great job explaining it. You just get so much more depth of understanding what people are talking about. So these interviews were about an hour long. They could be as short as 30 minutes, as long as 90 minutes. And sometimes it was one person and sometimes it's a couple in the household. And these are the parts where you're incorporating the information that you're getting from homeowners. Later on, and I'm not going to talk about it today, but later on, these are the parts where we're incorporating the expert information as well. So those two things are combined to create the tool. But today I'll just talk about the interviews for the homeowners. There's still a lot of data analysis to do, but this is just so far. So we're really just getting into it. First of all, we leveraged Project Itch in order to recruit. So they had to have completed the Itch survey, and then there was an Itch follow-up survey as well, which had more specifics. In yard control, they had to agree to be in contact for an interview. We did get an IRB approval for this as well. I think I had it on the last slide. They had to have a certain size property and they had to be available. And then we compared homes that were treated versus untreated for backyards. Okay, so as I said, 1 to 2 people per household. We had people that participated in Itch in 2023, those that were in 2024, and there were a few that had done it both years. Theoretically, there were 12 and 12, so we're 24 total. And here they are. The Itch sampling is the ones in black. So there were a lot of areas, and then the ones that we're calling scratch, itch and scratch interviews are in orange, and those that had management or no management. And by management, I mean, here we were talking about a pesticide, either synthetic or natural. So really not speaking at all to whether it was effective, but just that the homeowner made the decision to either themselves or professionally take an action for tick control. Okay, what we did find is once we went out and actually interviewed people, even though they had put on their Itch survey that they had not treated, and their Itch follow-up survey that they had not treated, there were 2 homes where they actually had been treating. So this was very interesting, right? What people considered treating. There was one person that thought they were treating their yard, but they were just mowing their lawn. So that term alone, we realized, is not as clear as you might think. And actually, this is one of the homes that had the discrepancy. So you can see we did end up considering them treated because it was about the decision they made and not necessarily about the efficacy of what was applied, which was, yeah, not very effective tools according to what we see today. Okay. All of the participants had had a tick-borne illness, either them or someone in their home. And many of them said that participating in itch didn't actually change their decision in the following, in either that year or in the future. But it was very helpful to get that information. So I thought that was interesting that they just flat out were like, no, it didn't really change my decision making. All right, so health seems to be a very big driver for individuals with this decision making of whether they're going to do a tick, any sort of tick control in their yard. So either protecting my family's health, my health, or my pet's health, I kind of lump those all in, and most of them were considering that. This was another interesting thing. I think the money I spent on tick control is a good value. There's not, and by the way, there's not a lot of differences between those that were untreated or treated for most of these. Again, we'll see. But what I saw here, I just wanted a few questions to say. Is the population in the interviews scratch about what we're seeing in the larger population of itch? And it seems to be somewhat similar, the difference between them. So that's good. All right, here for this one, what we did is we wanted to know, we had 4 potential objectives for them. Enjoyment of outdoor spaces, as I want to maximize my enjoyment of my yard. Cost, I don't want to, I want to minimize the amount of money I spend. Health, I don't want to get sick, right? That's a really important objective for me. And environment, and by this we mean like eco-friendly, ecosystem friendly, protecting the environment. And we gave them 20 poker chips, and we said divide them among these 4 objectives. So, and to see, how did they, it was a different than the kind of question where you're like, okay, is the environment important to you? 1 out of 5. It kind of becomes a little bit like Uber, like everybody gets 5 stars. Oh yeah, the environment's important. Oh yeah, health is important. Here we wanted them to actually make choices like, well, is it more important or less important than your health? And what we found was that basically there weren't a lot of huge differences here between like treated or untreated. We did find that in the environment category, that's the navy blue, there was a lot more variability. So like the standard deviation around those were much higher. So there were more extreme views potentially. And then cost actually was the only one. I don't know if I have it here. Oh yeah, cost was the only factor that did differ between the treated and the untreated, and not what you would expect. So the untreated homes, apparently they downgraded the importance of cost, probably because they knew they weren't spending any money. But those that were going to treat were more concerned about cost. So that's what I attributed to, but it was slightly, I don't know, cost significance there, 0.052. Okay, some other qualitative takeaways. So yeah, the health, environment, and enjoyment of outdoor spaces outweighed cost concerns. Households treating yards were more concerned about cost than untreated, and environmental concerns were the most variable. The biggest takeaway, I would say qualitatively, was the frustration that homeowners felt with the lack of clarity of what to do. So that there were so many options, they didn't know what to do. They'd get, they weren't really clear what the PCOs, what they were saying was accurate versus other sources. We really wanted clear messaging. And then another big outcome was that they were really concerned with efficacy. So that a lot of the recommendations are very broad, but it doesn't say, well, how effective will this be when I use it in my yard? And that's a really hard question to answer, but that's something they were really concerned with. So they wanted that to be an objective. I don't know if it makes sense as an objective so much as like a measure of one of your objectives, but that was one. And then there was an additional objective of, is it worth my time? Is it worth the effort, basically? And there were, it depends on your stage of life, kind of, and that's kind of anecdotally, but if there was somebody that had like young kids, they're like, well, my time is really valuable to me. And so, how much is this gonna cost me in terms of my time and not in terms of money? So that was a big thing. So the next phase that we're going to go into, we still have a lot of analysis to do. This is really just the start of the analysis of the homeowner interviews. But next we are trying to find experts for the expert elicitation. And we'll try to make an adaptive multi-criteria decision tool. And basically by adaptive, I mean that we have a very long list of alternative actions, potential actions. There's over 100 potential actions. And the recommendations will be tailored to the individual. So they'll go in and they'll start answering screening questions kind of like they did with itch. But if they have pets, then there's a number of alternatives that'll be included. And if they don't have pets, those will be eliminated. So that in that way, we can have, you know, the best options is that subset of potential alternatives that really applies to their situation, right? So if they live and their property is completely wooded, that's a different set of recommendations than if they live in a primarily suburban area and they don't have a lot of woods. So we want it to be tailored to the individual screening questions. Again, we added this minimized time commitment because time is, you know, they say time is money, but time is different than money. And so for different people, they're like, that's a kind of separate issue. And then we'll be gathering basically, and I want to make sure that this is clear, that the expert elicitation is about getting the best judgment of experts that are familiar in the area. So it's not about saying, well, the data are showing that this is the best choice, because we know that even that those data are from a very controlled experiment versus applications. This is about harnessing the power, leveraging all of our collective expertise. And it's saying, okay, for that, for a given alternative like tick tubes, how would that impact minimizing human health? How would it impact maximizing enjoyment of the outdoors? And we put actual numbers on that. And we are trying to get around 10 to 12 different experts. And then put it all together in a smart table and get out a bunch of weighted scores, and we'll get investment. Okay, this is our best, you know, here's the top 1, top 3, top 5 interventions that you could do. It'll look a lot prettier for people that are using it. But I think it's a good way of at least informing the decisions that people make. So again, you can have the score and you can pick out, this is all just hypothetical data. So none of these, don't look at this and say, oh, that's the best one. But it can provide users with the ranked list of alternatives based on their specific risk profile. And I like this idea of combining like, what do we know with what people actually care about? Because I think it is important that they get to choose how they want to live their lives and want to do. So, okay. So yes, if right now it's volunteer, but I do think we need to find a way to compensate people for their time. It's going to take a bit of time. But if you would reach out to me, I think that would be really excellent. Get started with that. All right, I want to thank everyone in our lab, especially Lexi and NEWVEC, and thank you for inviting me to this meeting today.
And then we have Alyssa from UMaine doing our first lightning talk.
Okay, well, thank you guys. I'll go ahead and get started. I'm Alyssa Ballman. I'm also a PhD student at the University of Maine. I work in the vector ecology lab under Dr. Ali Gardner. And today I'm going to be talking about the timber harvesting impacts on black-legged ticks. So the first project I want to talk about is a community science project we did a few years ago called the Maine Forest Tick Survey. And this was a 2-year study that we ran in 2020 and 2021. And this was a project where we recruited private forest landowners here in the state of Maine, or there in the state of Maine, I should say, that own between 5 and 1,000 wooded acres. And we had a huge response. We had over 300 people apply in the first year to be involved in this project. And of the people, we had a really high completion rate of over 83% of all our participants completed all aspects of the project, which is very labor intensive, which I'm going to get into just now. So all of our volunteers collected data 2 different ways. They filled out sort of extensive surveys that was asking different questions about their sort of attitudes and beliefs on ticks and tick-borne pathogens. They also went out into their forests and did a number of forest vegetation measurements. And then they filled out sort of extensive histories of their forest management practices on their land, including when was the last time they had a timber harvest. In addition to filling out all that information, which was already time consuming, they were also trained to actively collect ticks on their properties. So we trained them to use drag cloths to go out to these property places on their properties that they've had some sort of forest management, or in the case of some people never had any forest management. And then they dragged for ticks and submitted those ticks to us for ID. So I'm just going to very briefly talk about some of the results. So this is looking at the year since the last timber harvest. And so we have years, 0 to 5, 6 to 10, 11 to 20, 20 plus years since the timber harvest, and then never. And never is kind of a little dicey category, just because some people may have owned the property for a shorter period of time and they say, well, I've never had a timber harvest. But it doesn't mean there wasn't a timber harvest on the property at some point. And then we have the number of black-legged tick nymphs per collection period on those properties. And what you can see here is that there are significantly fewer black-legged tick nymphs on these properties that have been harvested anytime in the last 20 years, which we were sort of surprised. I feel like a lot of us, sort of understand immediately after timber harvest, there'd be fewer ticks, but it's interesting how long the effect lasted. And so that brings us to our current project that we're working on, which is a long-term timber harvesting project. And we're working out at a place called Fry Mountain Wildlife Management Area, seen here in the yellow box. And then here's a zoomed in picture where we're working. And as part of their wildlife management goals, they do timber harvesting in different parts of this area. So for example, we have our 4 sites here that are controls that are never have any timber harvesting. We have a large area here that they've clear cut that we have 4 samples we're sampling in. And then we have what we're calling patch cuts. These are 1 to 2 acre plots that have had a timber harvest. And so if we zoom in on these patch cuts, we're sampling kind of 2 different areas within these harvested areas. So we have the area in purple that has been harvested. So this is what some of them look like post-harvest. And then we're also sampling the ticks in that sort of boundary perimeter area to see how that also is impacted by the timber harvesting as well. And so, like I said, this is a long-term study. We're currently on year 4 of 20. So check back in 16 years for some results. But like I said, we're doing the tick dragging as well as doing a number of vegetation measurements, including looking at understory composition, canopy closure, different leaf litter characteristics, as well as microclimate, looking at temperature and humidity in these areas. And so we're really trying to see like how this is changing over time. And we're looking at it over a pretty long time span to see just how this changes and how that impacts the tick populations. So with that, I just want to thank our funding sources, and I want to thank all the amazing volunteer citizen scientists that we had that worked on this project.
Hi, my name is Mike Galley. I'm also from Allie Gardner's lab, and I want to give everyone sort of a report on how itch went for the summer of 2024 and some of what we found. So in Maine, we're approaching itch in sort of a 3-year block, and our main goals for itch right now are to look at the landscape features and try and identify how those could predict tick density. And we're also trying to test the effectiveness of beta-cyfluthrin as a tick control pesticide. Now, Maine's a little bit weird. We're a lot different than other places in New England. One of the reasons is that our properties are really big. Our median property is larger than 5 acres. Our largest was over 100 acres. And Maine's tick densities are not uniform over the state. As you can see, this is ticks that were sent into UMaine Extension, and that's just about where our study sites are. So we're really studying the edge of where real dense tick populations are in Maine. Last year, we sampled 40 residential properties, and for those who really only did the landscape features, this year we upped it to 57 properties, and this is when this year's the first year we've started the spraying. None of our properties previously were treated for any kind of tick control for the past 3 years, and the reason that we wanted completely clean properties is because we did the treatments ourselves. At Maine, we are registered as pest control professionals, and we were able to actually spray Tempo SC Ultra, which is an off-the-shelf product that uses beta-cyfluthrin as its active ingredient. Beta-cyfluthrin isn't the most common tick pesticide in Maine, that's bifenthrin, but we wanted this our itch work to be future focused, and upcoming PFAS legislation means that bifenthrin might not be as available in Maine in the future. So we applied it to 27 different properties, and we only treated each property once in mid-June. We were over the summer able to get over 1,000 ticks from all of our properties, and the treated properties had about 50% less ticks on them than the untreated properties. And the spot we really noticed that treatment take effect was in zones A and B, that orange and yellow zone. Both of those reduced ticks densities by just about 70%. The other thing we noticed over the time scale of the summer is that where untreated properties had this big jump in black-legged tick nymphs in early to mid-July, treated properties didn't get that jump. To sort of orient this graph, each of these dots are the average of all the properties we sampled that day. So each of these dots is the average of like 3 to 4 properties that we sampled that day. And you can see that just mid-July, we just really jumped up in the number of ticks. So because of sort of not having that big jump, we're really excited. We think the product might be working, and I'll be happy to take any questions.
All right, hello, everybody. My name is Sandra. I'm a master's student at Western Connecticut State University. I'm working with Dr. Nita Connolly. And for my thesis, I'm actually looking at how mowing impacts black-legged ticks in people's backyards. And today I'm going to share a little bit of what I've been doing. So to recap, mowing is currently being recommended as a possible tick control method or tick prevention method in people's backyards. The idea is that you cut back the grass, let the sun come in, decrease humidity, and make the area inhospitable for ticks. However, there's not a lot of research that actually supports this, and we are seeing some homeowners moving towards mowing their backyards less in a way to help with conservation efforts such as No Mow May. And so the objective of the study was to evaluate how lawn mowing frequency affects black-legged nymph behavior at the backyard forest edge. To do this, we created a randomized block study design that had 9 enclosures divided into 3 different blocks. Each of these blocks had one of our mowing treatments, whether it was mowed weekly, mowed every 2 weeks, or not mowed at all. Each of these enclosures was 4 meters by 1 meter in dimensions, and they were placed in the backyard so that 3 meters would be in the lawn and 1 meter would be in the forest. We then added Hubble data loggers to keep track of the microclimates, and we infested the forest area with 300 lab-reared black-legged nymphs 1 week before the start of the study. Then during the study, the area was sampled on a weekly basis for 8 weeks by flagging. So these are what enclosures actually ended up looking like, with these tall sides that kind of help keep the ticks in, and the crop cage tops that help keep the wildlife out. What did we find? Well, we found that mowing did not significantly change the number of questing ticks. Here in this graph, you can see, hold on. This is the mean number of ticks found in the lawn over all 8 weeks. And these are different treatment types, the mowed weekly, mowed every other week, and not mowed. And as you can probably tell by the graphs, it was not significantly different. So when trying to figure out, well, why didn't this affect the ticks? We looked at our microclimate data, right? So we know from previous research that there's a suspected minimum humidity threshold that ticks need to survive. And we think they suspected that it was between 75 and 82% relative humidity that they need. When we look at our data, we see this is the mean weekly humidity, mean weekly lawn humidity the week before the sampling date. These are all our sampling dates, and the light yellow is our mowed weekly. The middle bar is going to be the mowed every other week. The dark is the not mowed at all. And as you can see, throughout most of the 8 weeks, the humidity stayed pretty high, well above 82%, with the exception of week 4, where when it dropped down, it really only dropped down to 80% plus or minus 2%. And so we also wanted to think of, well, why is the stain so humid? And some of the factors that we think may have impacted that include, we chose a mowing height of 3 inches. We also did not remove any lawn clippings or any fallen leaves. And there was a significant amount of tree overhang from the adjacent forest. And we think that these three things actually created enough shade to keep that humidity in regardless of the mowing. And so some final thoughts. Mowing a lawn did not reduce questionings in this study, right? So what this really means is that regardless of the mowing frequency, homeowners are still going to be at high risk of tick encounters at those forest edge spaces. And so it's worth it to consider adding additional humidity reduction strategies to the mow your lawn frequently recommendations, such as trimming back your canopy, picking up all the leftover lawn clippings, or even maybe mowing to a certain height that's shorter and allows more of that sun.
Hello, everyone. My name is Fatma, and I'll be presenting some more results from the ITCH Knowledge, Attitudes, and Practices Survey. Dr. Andrew Lover touched upon it in his presentation. We'll be going a little bit deeper into some other results we looked at. And I want to start off by acknowledging the folks in the room that created the survey and drove kind of recruitment and thus the results I'm showing you here. So briefly, to overview what I'm going to talk about, we're going to talk about some general characteristics of the participants in the survey, moving on into the prevalence of household level vector control interventions, and then looking at a model to predict any household level vector control based on state, yard size, and yard type, and then any next steps for analyses. So looking at yard sizes across the region, looking at by state, we'll see here that at least for the southern states, you'll see a lot of participants, having up to an acre or 1 to 3 acres. And moving up north, you'll see more participants will have more than 5 acres, which is not surprising, as many of you have said today, but it does kind of quantify, puts it on paper, these differences in the yard sizes. And then looking at yard type, most of the participants, as you can see, answered based on this picture right here, hey, does your property yard type fit into A, B, C, or D? Thinking Dr. Connolly, Dr. Mather, and kind of broadly classifying that, most of them fit into A and C here. And again, it kind of shows, who's responding to the survey and helps us quantify what the yard types look like and able to inform like policy or any vector control programs by looking at any levels of concerns and attaching it to that as well. Looking at some prevalence of the household level vector control interventions from the survey, we'll see it's important to note here that in 2023 and 2024, the question was changed a little bit. So in 2023, it was asked, hey, did you use any vector control for ticks or mosquitoes? And then in 2024, it's kind of split up as depicted here. Something to note here for commercial pesticide application, there is a little bit of a jump between 2023 and 2024, which is expected as labs were recruiting participants who had treatments on their properties. And then it was surprising to see so many more people reporting to use tick tubes, 10 and 11%. So that might actually just be representative of who's taking the survey and responding. And this is a multivariable model looking at any household level vector control, yes or no, adjusting for household income and for a survey year across states, yard size, and yard types. So we'll see across states, Vermont and Maine properties are actually less likely to use vector control than Rhode Island as the reference group here. Yard sizes, those with a greater than 5 acres are less likely to use vector control, household level vector control up than properties with up to an acre. And then yard types, those who are in B and D are less likely to use vector control than yard type A as reference group. And so for next steps for further analyses, we're going to look at levels of concern about risk for uptick or mosquito-borne diseases with residential vector control interventions, one being yes, no, the other being lowest, medium, highest total vector control. And then also looking at property characteristics like geographic region and neighborhood classification, urban versus suburban versus rural on those outcomes as well. Also looking at willingness to pay and then sub-analyses of each state. Hello, I'm Veronica Bobstill. I work in Dr. Lover's lab as a seasonal surveillance technician manager. And these are vegetation analyses at long-term tick surveillance sites. So vegetation is largely the way that ticks interface with their hosts. Understanding more about vegetation may teach us more about their habitats and their life cycles. So we have three main goals. One, to quantify the interannual variability at a lot of different ecological sites. Two, to create a protocol that's comparable to collaborators. And also to conduct these analyses and our plot placement. So our typical tick collection plots are 40 by 40 meters, which is pretty huge. For this vegetation analysis, we only sampled the vegetation at a subplot every 20 meters, which, as you see here, these little blue blocks are the subplots, and they are 3 by 3 meters, comparable to what is being done in Vermont. And we have a total of 8 subplots per tick surveillance plot. And in the picture, there's James placing the plot, and we use a 3 by 3, a 3 meter PVC pipe to make it way easier to place the edges. Vegetation metrics. So we looked at leaf litter composition, invasive species density at a 0 to 3 scale, 0 being none present, 3 being pretty filled. As you can see in the pictures, we did the same thing with understory vegetation coverage. This included invasive and non-invasive plants. We looked for the tallest plant and shrub in the plot and quantified their height on a scale of 3. And we did presence and absence of select features, including mass species, tall grasses, and berry species, as all of these could lead to different hosts being around or a positive habitat feature for ticks. We also looked for leaf litter depth at 2 locations, one on each side of the subplot. We collected basal area and used hobo loggers to collect temperature and relative humidity, and searched for canopy coverage using a GRS densitometer, which that's the view through it. You kind of look through the crosshairs at a little point, and we looked at 8 different spots per plot. Takeaways: it's important to balance collecting qualitative and quantitative vegetation metrics when working with limited time. So if everything was quantitative, it wouldn't be possible to get this work done in a timely manner. And we also could not use the exact same sampling methods due to different habitat and, again, staffing. And it was really beneficial to create tools to make sampling simpler. And I have physical copies of this protocol that are happy to share with anybody if they'd like to look it over. I'd like to thank my field crew and collaborators and the Lover Lab.
Hi everyone, I'm Johanna Ravenhurst. I'm a PhD candidate in epidemiology working in Andrew Lover's lab. And I'm going to be talking today about Project Listen, investigating the experiences and challenges of public health and vector control professionals across New England. So the overarching research question for this study is what could NEWVEC do with our resources to help with integrated vector control and surveillance across New England? And within that research question, what are the barriers and facilitators to achieving the goals of this work in our region? And what are the main organizational and logistic barriers? So Dr. Lover already went over our sampling plan a bit in his talk this morning. So I'll just briefly say that so far, we are partway through this study. We have collected data in what we call strata 1 of our recruitment plan, our sampling plan for this study. So the preliminary results that I'll be presenting today are based off of an in-depth analysis of the 12 individual interviews that were conducted amongst that group, and we are currently conducting data collection in the subsequent groups. For this study, we're using an inductive coding process, meaning that our themes are developed from what participants are telling us. So we're looking at detail in at the data we collected through our interviews, and then we developed this list of preliminary themes. And just to give you an idea of the code book, this is one theme here, and all of the codes that are under that theme, and then the definitions that we use to apply those codes. One example of a theme is collaboration and relationships, and interagency coordination is under there. So we're hearing from people that open dialogue is key to effective interagency coordination, and weekly or bi-weekly calls are really important. There's also acknowledgement that mosquitoes and ticks aren't constrained by state borders. And so we need a regional approach to surveillance and control. And the context here was in terms of collaboration and communication amongst professionals working across the region. Educating the public is a code that came up under communication and influence. We've been hearing about many innovative methods for educating members of the community about how to protect themselves. And some, even as the person said in this quote, are even placing educating the public at a higher value above their own work, because they can't be everywhere at once. And so if they're able to educate people to protect themselves, then they can mitigate their own risks from vector-borne diseases. Under emotional and psychological impact, stress and pressure. So the people, this is the people working boots on the ground doing control and surveillance work. They have an increased workload during the mosquito and tick season. And they've been sharing, like, especially last year, for instance, it was a busy year. They were kind of burnt out, exhausted at the end of the year. And then it just didn't end. Tripoli positive pools started popping up, emu cases and, all these animal cases coming up. And so the work just didn't end when they were just ready for it to end. And somebody shared that they felt they couldn't even get up from the microscope because they needed to keep sorting mosquitoes and get them to the state to be tested. They've been talking about industry changes and adapting to industry changes, and how there's been an increase in competition in the field. And many people have noted there's these private companies that have been around for a while. Maybe they're family owned. Maybe they have, stronger connections with the community, have built a reputation. And then in the past 5 to 10 years, we've had more opportunistic pest control companies come in who maybe have different standards for the work that they do and fewer connections with the community. So it's just something they've been noticing. So some of our preliminary results include, I've already gone over a few of the things that have been going well in terms of regular communication and collaboration, team support, prioritizing staff safety, and this emphasis on education and knowledge sharing, not only with members of the public, but among professionals working in the field. Some of the frustrations and barriers that have come up are, public misunderstanding of the work, finding staff with specialized training, such as being able to ID mosquitoes, staff turnover, both internally and then also at other agencies, leaving a gap in institutional knowledge that somebody needs to fill. And then just gaps in information or communication. This is just a short overview of what we're seeing so far, but we do have a work in progress presentation scheduled for November 6th. We'll be talking about this quite a bit more. And I just wanted to give a huge shout out to our team at the Center for Program Evaluation here at UMass in the School of Public Health.
It's great to see everyone here. We work together as a team. My name is Guang. I'm going to report a new discovery. We found a new spotted fever group, Rickettsia, in rabbit tick, Haemophilus liparis plastrus in Maine. So we all know Rickettsia can transmit disease to human or animals during the blood feeding by infected ticks, mice, lice, or fleas. Based on the phylogenetic analysis, they are at least 4 groups. The major the largest group is spotted fever group. Within those group, there's the most lethal disease, Rocky Mountain spotted fever, caused by the Rickettsia rickettii. This is most lethal tick-borne disease in the United States. So I think up to today, there are 5 Rickettsia groups found in the United States. Rickettsia parkii, usually we found this in Southeast United States, but in this year, Rickettsia parkii and its vector tick, Gulf Coast ticks, moved to Connecticut. So I think we'll find this next year probably in Massachusetts. Rocky Mountain tick fever was transmitted by, usually we think it's transmitted by dermocenter ticks, but recent study shows brown dog tick is probably the most important vectors compared to dermocenters. Cape Cod used to be the hot spot for Rocky Mountain spotted fever, but it's very, very rare in New England now. Most cases found in the middle of the United States. The last 2, Pacific Coast tick fever was transmitted by a different tick species only found in California. And last month, a new Rickettsia species, CA6269, they proposed a new name called Rickettsia lenii, was reported in the EID journal. It was transmitted by rabbit ticks. So we test a lot of ticks. In those 2 years, we test over 6,000 each ticks. Here is our workflow. So once we receive the tick, we first ID it using the morphology characters. We divide it in 2 groups, exotic tick and non-exotic ticks. So we use exotic package, test exotic ticks, and here is the example for non-exotic ticks. For each tick, we test 3 different packages. The first package contains all the species tests. The second one tests, we call it NIT1, tests Rickettsia borrelia. The last one, we test tularemia, anaplasma, and the Rickettsia. So each ticks, we run 3 tests. So last year, we received 3 ticks. At the beginning, we received 3 ticks from Maine. It was kind of surprised. We found 1 Rickettsia positive, Rickettsia positive. It's really a surprise because we test, since 2006, we test over maybe more than 80,000 ticks. We didn't even find even 1 Rickettsia positive. So this was our first Rickettsia positive. We report this to Ali. Then Ali sent another 190 ticks. I believe 10 of them we tested were positive. This year, she submitted more rabbit ticks. In total, you can see this number, 18 of 298 rabbit ticks collected from Maine positive. The rate is pretty high. It's 6% of them are positive. I have to say, why Maine group collects those many rabbit ticks, I don't know. So we got a small number from other states. That's a lot of rabbit tick from Maine. So once we test the positive, we want to know what kind of species, because Rickettsia is very, very similar. The sequence is very, very similar. Although this assay that's a CDC assay, says that's Rickettsia. We want to confirm it. We use the multi-local sequence typing method, MLST method. We use 4 genes, outer membrane protein A, B, and the surface cell antigen 4 and GAL-A. We PCR those 4 genes, purify it, sequence that, then align with available genome sequence. We build a phylogenetic tree and found the top one, the right color is the rickettsia we found from Maine. What will we know from this trait? The first we know this is the new rickettsia. And we know probably this rickettsia will cause some kind of human disease because you can see it grouped together with the Rocky Mountain spotted fever pathogen Rickettsia rickettsii and 2 human pathogen from California. So it's very likely that's pathogenic. And we know it can transmit from adult female to next generation because we found this pathogen in larva, nymph, and adult, all the stages. It raises more questions. We need more studies. First is, will this kind of tick transmit this new rickettsia to human? I guess yes, because this tick is a 3-host tick. It's mainly fed on rabbit, but it seldomly fed on birds and small animals. It does still feed on human, but very, very rare case. So the recent paper found, okay, 2 patients, they encounter rabbit ticks, but the frequency is very low. In 20 years, they have 2 patients. This very interesting thing is, 1969, 13 rickettsias Rocky Mountain spotted fever case was reported from Cape Cod. But if you look at the time, it's from April to June. We thought this was transmitted by dog ticks. It's very hard to find dog ticks at this time. And from this 55 years, nobody even isolated or detect rickettsia from demo centers. So maybe, I guess, it's probably that's a missing link here. So we need to do more study for the rabbit tick and this new rickettsia. We don't know, right now we don't know the host. I think Leslie told me this morning, all the ticks we tested are all the flag ticks. We don't know the host. Definitely we need more studies. This is a teamwork. I want to thank for the main group and the UMass group. And Steve, thank you, Steve.
Those are very nice presentations. I don't really have data to show you, so consider this more of like a work in progress. So yes, I titled the talk Innovating the Use of Plants Beyond the Steel Archetype, and sort of just want to discuss like how we get into this line of thought. So I originally started in Steve's lab. I worked with Pat doing just basic mouse genetics that were effective. And then I was looking for a reason to stay in the lab, and I was hired to continue a DoD project, which looked to see if we can apply the use of spatial repellents to ticks. And this is sort of a new idea for ticks, because the main reason that we use spatial repellents is for insects, and that induces either an excital repellency response or some kind of vaporized response that either deters mosquitoes from landing. But the main thing is they're not actually touching the insect itself. They're sort of perceiving the vapor that's. And so one of the challenges to actually applying spatial repellency in ticks was developing an assay to determine whether it's effective. Ticks do not behave like mosquitoes. They do not behave as you predict. That's been sort of one of the main challenges. How do we measure this repellency and how can we apply it? And sort of taking this step back, thinking about a bigger picture of how to use repellents. If we look at the process by which a a tick, as it emerges from leaf litter, it goes through the host seeking process from the beginning to when it gets unfolded. It comes out of the leaf litter, and there's a series of interactions, whether it's as it's questing, the interaction between when it comes to your host, and then once it goes onto the host, and then a series of smaller behaviors before it gets being molted. One of our ideas is to actually define these behaviors, define these checkpoints that a tick has to cross as it goes through this life cycle, and see if there's a way that we can manipulate these behaviors with repellents. Sort of as almost a substitute for the synergism effect with the parasites, and see if there's a way that we can use some of the molecules that we've been developing that either show effects based on a tick won't cross from point A to point B. Can we apply that to sort of non-infections? And so this sort of line of thought is taking us from, think of these ideas of how we can use repellents in other than just spraying either skin or treated clothing, find these behaviors that a tick needs to go through, translate those behaviors into quantitative measures, standardize the assays that are used to assess these measures, and then we can sort of piece together this algorithm of we test a molecule with assays A and B, and based on the results of those assays, those could translate into different specific use cases. One of the molecules that we've worked with is NEWVEC, which Thomas worked with as well. And in the lab, what we see is it was originally thought of as a lethal molecule. So it kills ticks at very low concentrations. And at concentrations, I've only gone as low as 0.0029% in the lab. And this is with adults. The ticks actually won't stay on a filter paper that's treated long enough to produce a lethal effect. So the ticks are being repelled at an even lower concentration. And one of the reasons that this is meaningful is there's a study where comparing emulsified butyl formulations with encapsulated butyl formulations. And if you treat an environment with encapsulated butyl, the molecule actually persists in the environment for several months at roughly the same concentrations that it was treated at. And the phenomenon that you see is when you spray the environment and you flag the environment, you get almost no ticks, if any. And the question is the effect that we're seeing, the ticks that are there are being killed, the ticks that are coming onto this sprayed environment are also being killed, or are the new ticks that would be colonizing this environment being repelled before they can get there? And so the importance of understanding that is if that is the case, then that's justification for the use of environmental measures and wondering whether you can either use these dual action molecules for that role or use these molecules in combination with lethal effects. And sort of this extends beyond that too. So that could affect the questing stage. But what about like on-host measures? So for tsetse flies, there's a very effective collar that was developed that emits spatial repellents that actually prevents tsetse flies from causing disease in cattle. And so that works with the phenomenon of using non-host semiochemicals. So pretty much masking the scent of a good host for a tick with the sense of a bad host for a tick that the tick perceives and then host. And so this has actually been applied very briefly in dogs using some vertebrate-limited almonds against brown dog ticks. So another question is, can we take that phenomenon, use these non-host semiochemicals in either as spatial repellents or contact repellents, either treat reproductive hosts or treat environments with it? Can they be applied to fabrics, either treated fabrics or manipulate fabrics that are impregnated with them? And so different use cases, the idea is take a molecule, see where it fits into affecting the tick's host life cycle, and then find use cases for those molecules. And so there's a number of other questions that we can ask kind of like based off of this. So in some of the blood meal analyses, what we see is that ticks are feeding and that they're dropping off and finding new hosts to feed. They're being groomed off, or they're falling off. And so some of the papers we're seeing are 5 to 10% of ticks are having these incomplete blood meals that have DNA from 2 different hosts. And one of the reasons that this is important is we actually need to figure out if we were to use repellents, we're not killing the tick, so it could fall off whatever host or whatever surface. How likely is it that that tick can actually find a second blood meal? How efficient are ticks at actually doing that? Because if a tick that is repelled 5 to 10% of the time can actually find another untreated host, say it's like a reproductive host, the maximum efficacy that we would be able to get from sort of trying to fill in the role of the parasite would be 5 to 10% lower than the maximum efficacy of the comparable molecule. And so that's an important question to answer. Are we actually seeing 5 to 10%, or is that a little bit inflated either based on some of the DNA analyses that are not efficient? And another thing to look at is one of the, I guess, qualities that you would look for in a repellent that might be better than the parasite is that you lose the selection for resistance that you might see with parasites. So if we can substitute, especially with like politics in certain areas, if we can find these molecules that work for either treating environments or treating some of these animals with these repellents, what do the effects look like in terms of sustainability? So that's what I want to discuss. Sort of, we're kind of piecing together this pipeline of defining the behaviors that a tick has to go through from emerging out of leaf litter to finding a host. What behaviors can we manipulate? How effective are these molecules at actually manipulating them? And then kind of set the stage for like kind of poking the system with different environmental conditions. So can we actually get repellents to work and apply them? But what happens in the sense of like heat waves? Are the repellents less effective because ticks will be more desperate in heat waves, or like environment predictions? And so trying to figure out the system. That's all I wanted to discuss today, but hopefully in the next couple of months we'll have some actual data.